My name is Efrem Lim. I'm Associate Professor at Arizona State University at the Biodesign Institute. So I grew up in Singapore, and in 2003 we had the SARS epidemic. That was a novel virus that really caused a lot of panic and fear. We did not know anything about that, and that kind of triggered my interest in science and led me into microbiology. In infectious diseases, viruses are overwhelmingly contributing to the disease burden. One of the interesting things is that the viruses evolve over time, and that causes them to have different changes, causes severe disease, or different ways that we have to adapt to treat them, create vaccines to update them, and so that led me to be interested in the genomics of viruses, the genetic code, and how they evolve over time. And those mutations that come out from the evolution have a very large consequence on how the virus then behaves on the population. One of the main questions that we have of the human virome is that many of the viruses that we harbor are not well understood and not characterized. And so there's a basic gap in this knowledge about what viruses are present and then what are they doing. And so that's where we're using genomics to study and identify the viruses in the human body, in different compartments of the body, asking how are they playing a role in our health, what constitutes a healthy virome, and then also what's their role when it comes to disease. And so the minute I heard about this new coronavirus in 2020, and it was related to SARS, it was a natural fit for me to pivot my lab to study that. And so we've been sequencing this virus ever since the first start of the pandemic. We've sequenced more than 100,000 cases in Arizona, and we are responsible for keeping track of how the virus is evolving in our communities. So in the evolution of SARS-CoV-2, one of the reasons we're tracking this virus closely is because mutations in the spike protein can affect monoclonal antibody therapeutics that are used in the hospital. That means that if the virus evolves a mutation there, some of these antibodies that we give in the hospitals will not work. It will not save the patient's life. So rather than thinking of them as these different variants and so forth, clinically at the bedside, we are really interested in this fundamental question of, will this antibody save the person's life? And so that's where digital PCR comes in. Using the Absolute Q digital PCR system, we've developed a suite of assays now that can target specific mutations in the spike protein of SARS-CoV-2. Absolute Q's digital PCR is the next generation of PCRs. Yes, there is real-time PCR, but you never get the specificity of a single point mutation. Absolute Q digital PCR can do that very accurately. If it's just one nucleotide change versus another, it can exquisitely detect that change. That's the reason why we went with that digital PCR. So our research directly can translate to all these other infectious disease viruses. In fact, what I'm really happy about this pandemic is that we've shown and demonstrated that the power of genomics to understand viruses and infectious disease. Before this, no one was really interested in sequencing viruses or learning from the genomic sequences of pathogens. We've showed the power of that, and so that's where I feel that now that we've shown this with SARS-CoV-2, we would definitely continue that with influenza of other viruses and show that that's how we can change public health in the future. I think an important aspect for scientists is to be open-minded and to be daring, because research is at the frontier of knowledge, where many of the things that we are doing has not been done before, and you have to take that bold step forward to just give it a shot and try and see. Sometimes you'll fail, the experiment fails, and it's disappointing, but there will be times where you learn something remarkably new that no one else in the world knew before you. And so that's the attraction to science, is that you're really at the frontier of knowledge.